Hello everyone. Today we will discuss our new chapter of the human physiology in summary, right? That is body fluids and circulation. As its name suggests, body fluids, that means in that body fluids the blood and the lymph comes under it, right? And with circulation, that is the whole circulatory system. You can see the structure of heart, right? The blood vessels, nerve vessels, all these vessels, right? So we will study all these things about in body fluids and circulation, right? As we know that in body fluids there are two major fluids, that is blood and the lymph. Clear? Blood and the lymph. Blood is the major transporting fluid. Blood is the major transporting fluid. Right? Blood is colorless, blood is colored because it is red in appearance, and lymph is the colorless. When we talk about the lymph, it is formed by the filtering of the blood as well, and again mixed with the blood, right? So blood is major transporting fluid. When we define it, it is opaque, turbid, sticky fluid, connective tissue, right? Blood is the opaque, turbid and sticky fluid connective tissue. It is turbid, opaque and sticky fluid connective tissue. Opaque, that means we can say we cannot see across the blood, right? It's not transparent actually. That's why the word opaque is used here. Sticky, it's stick like that, right? And through the blood, all the nutrients are transporting in the blood. The hormones is transported through the blood, right? It also helps to for the distribution of the heat uniformly in all over the body, right? The oxygen and carbon dioxide is transportation occur through the blood, right? So blood, that's why the blood is major transporting fluid. In blood there are three major things which is present, and one more thing, blood is the fiberless. It is the fiberless, right? Blood is the fiberless. That's why it is fluid in consistency, right? Fluid consistency, liquid in appearance, right? In blood there is blood plasma, which forms the 55 to 60% of the blood part, right? In NCRT clearly mentioned 55% of the blood plasma is present in blood, right? Second one is the blood cells, right? And so all these things are present in the blood. When we talk about plasma, it is pale yellow, clear colorless, transparent fluid, right? It is clear colorless, that is pale yellowish in appearance. Actually it is pale yellowish in appearance, right? Aqueous fluid, and pH is 7.4. pH is 7.4, and due to the presence of it, the pH of pH of blood is also 7.4, right? when we take its range, that is 7.1 to 8.5, but on a when we take a standard criteria, that is 7.4, right? Plasma has various components. Plasma plays a major role in also in protein synthesis, in providing the defense mechanism of the body, right? There are three major classes. Mainly it consists of 55% of the blood, right, in which 90 to 92% water is there, right? 90 to 92% water is there, and what will and in 8%, other 8% rest of the substances like the immunoglobulins, right, like the immunoglobulins which secretes the antibodies, right? Defense mechanisms or defense substances like the lysozyme and the properdin, right? Lysozyme and the properdin. Minerals, vitamins, enzymes, all these kinds of, including the proteins, and there are three major class of proteins present in the plasma, which are known as the plasma proteins, and they have very important role actually, right? That is the serum albumin, serum globulin, and the fibrinogen. It helps to maintain the osmotic gradient. It helps to maintain the osmotic gradient. Globulin secretes the antibodies. And fibrinogen is the clotting factor. Right? When we talk about the fibrinogen, we know that it is the clotting factor, blood clotting factor 1, right, which helps in the coagulation of the blood, Other than that, some minerals are also present in the plasma, like the sodium, potassium, chloride, right, all these things. But when we solve the questions, the question is the basic buffer of the body is, or the basic buffer is sodium bicarbonate, right? This is the question of pre-medical. The basic buffer is the sodium bicarbonate. Other than that, all other things also maintain the buffer of the blood, that is the hemoglobin, right, indirectly, albumin, right, all these things also maintains the buffer of the body. Other than that, some nutrients is also present in the plasma, like the amino acids, glucose, right, amino acids, glucose, fructose, all these nutrients are also present in the blood. Some excretory waste products like the urea, uric acid, creatinine, right, all these things are present in the plasma. Clear? So this is all about the plasma. Now come to the formed elements, right? Formed elements, there are three formed elements. There are three formed elements, that is the RBCs, which is also known as the erythrocytes, WBCs, the leukocytes, and the platelets, that is thrombocytes, right? RBC, WBC and the platelets, that is erythrocytes, leukocytes and the thrombocytes, right? Now come to the when we talk about the RBCs, that is the red blood cells, which is also known as the erythrocytes, clear? These are first question, these are the most abundant blood cells. Why they are called most abundant? Because RBCs, the number of RBC, if we talk about only NCRT data, then NCRT says that the normal range of RBC is 5 to 5.5 million per cubic mm of blood in males and females both, right? But when we go through the medical data, then there is little bit variation. In females, the RBCs is 4 to 4.5 million per cubic mm of blood, and in males it is 5 to 5.5 million per cubic mm of blood, right? So, but because we follow the NCRT data, so it's 5 to 5.5 million per cubic mm of blood, right? That is in lakhs, it is 50 to 55 lakhs, right, per cubic mm of blood. The formation of RBC is known as the erythropoiesis. For the erythropoiesis, there is hormone is necessary, that is called the erythropoietin, right? The hormone which is necessary for the formation of RBCs, that is the erythropoietin, clear? That erythropoietin hormone is necessary for the erythropoiesis, and that erythropoietin hormone is secreted by the kidney, mainly 85%, and the liver, right, 15%, clear? So in RBCs, all these things. Now come to the next. Right, we already know that now the formation of RBC is called the erythropoiesis, right? The hormone erythropoietin is necessary for the formation of RBCs, right? Erythropoiesis. And the site of erythropoiesis is, when we talk about the site of erythropoiesis, from third week to third month, from third month to 5th month and 5th month onwards yolk sac, liver and spleen, and the bone marrow. Right? So what will happen now? The question in NEET 2015 and 16, right? The question is asked in embryo, the RBC's formation occurrence or the main site for the erythropoiesis is, if it is not mentioned in adults or in children, so consider it as child, right, as fetus or the newborn, right, as fetus, and mark the answer liver, right. Don't mark at that time the bone marrow. Clear? Now what will happen? We know that the RBCs, the structure of RBCs in the humans, RBCs per day, per day or per second 2.4 million RBCs are formed in our body, right. When we talk about the structure of RBCs, it's circular, biconcave, non-nucleated, or we can say the enucleated, right, non-nucleated or the enucleated, right. Now what will happen? That is in case of humans, but in there are some exceptional cases also there in mammals, right. Most of the mammals have this kind of, but in exceptional case are there. Exception is the camel and llama, right. Camel and llama. Oval, biconvex and nucleated, right. They have the oval, biconvex and nucleated. Clear? So this is the structure of RBCs, and the RBCs is covered by a plasma membrane. So its plasma membrane is known as the Donans membrane. Plasma membrane of RBCs, right. Clear? It is permeable for the chloride and bicarbonates and impermeable for the sodium and potassium ion, and that phenomenon is called the Donans phenomenon. That phenomenon is called the Donans phenomenon, right. And when we talk about the lifespan of RBCs, right, in humans, 120 days. In rabbit, 80 days. In frog, 100 days. Clear? Now, after the completion of its lifespan, when RBCs get disintegrate, so these RBCs destroyed in liver, and after that they buried, completely destruction occurs in the spleen, right? That's why that spleen is known as the graveyard of RBCs, right? Now, what will happen when RBC is destroyed in that liver, right? So, RBC carries, actually, why RBCs is red in appearance? So, RBCs carry the hemoglobin, actually, right? RBCs carry the hemoglobin, and hemoglobin is what? That is iron containing cytochrome respiratory pigment, right? Iron containing cytochrome respiratory pigment. That hemoglobin, even when RBC destroyed, then hemoglobin destroyed, and when hemoglobin destroyed, it gives that hemoglobin is made up of 5% of heme and 95% of the globin, right? It is made up of 5% of heme and 95% of globin. When that hemoglobin get destroyed, right, it gives three, that is ferritin, three products: ferritin, bilirubin, biliverdin, right? Ferritin, bilirubin, biliverdin. So all this ferritin is reused in body, right? This ferritin is reused in body, and that bilirubin and biliverdin are the excretory waste products of the liver, right? These are the excretory waste products of the liver. Clear? So in that manner, that hemoglobin, when RBC is destroyed, then hemoglobin destroyed, and hemoglobin destroyed, then these byproducts formed, right? If the liver, these bilirubin and biliverdin is metabolized by the liver, if the liver is does not function properly, then what will happen? The amount of bilirubin and biliverdin increased and causes increased amount, causes the jaundice, right? Increased amount of bilirubin and biliverdin causes the jaundice. That's why after the malaria there is more risk of the jaundice, right? After the malaria there is more risk of occurrence of the jaundice. Clear? So simply RBCs destroy hemoglobin, RBCs destroyed, then hemoglobin destroys, then what will happen? All these things gets destroyed, right? Now hemoglobin its oxygen binding capacity is 1.34 ml. per gram, right? Oxygen binding capacity is 1.34 ml per gram. So what will happen now? When normal range of hemoglobin in our body is, or we can say if we go through the NCRT, so NCRT says the amount of normal hemoglobin is 12 to 16 gram per DL or per 100 ml of blood, right? Clear? So hemoglobin, that globin is made up of 4 polypeptide chains, that is globin. These are the 4 hemes which is attached with the globin, right? And it carries the 1 iron, 1 oxygen, 1 iron, 1 oxygen, right? So molecule of hemoglobin, 1 molecule of hemoglobin can carry 4 molecules of oxygen, right? And they are globin is made up of 4 polypeptide chains, right? 2 alpha and in adults alpha 2 beta 2, in fetus alpha 2 and gamma 2. Clear? So this is all about the hemoglobin, right? Now what will happen? See, there is a method that is the hemocytometer, right? Hemocytometer, hemo that is related to the blood, cyto that is related to the cell, meter that is the measurement, right? So what does it mean? It is an instrument, right? It is an instrument through which all the blood cells, the number of WBCs and RBCs can be estimated, or we can count the number of RBCs and WBCs through the hemocytometer, right? Come to the Raulex formation. Raulex formation. What will happen in Raulex formation? Due to the discoidal shape of the RBCs, its unique shape of RBCs and increased amount of fibrinogen, sometimes the RBCs get piled up to each other, right? And by piled up to each other, they form a specific structure like that, right? And that is known as the Rolex formation. This is called the Rolex formation. Clear? Fibrinogen favors the Rolex formation, and it is excessive in it is just due to the excessive amount of the fibrinogen and the or we can say the high plasma protein, because fibrinogen is the high plasma protein, right? So this is the Rolex formation. Now what will happen if the amount of RBC or the WBC is the amount of RBCs or the amount of RBCs or hemoglobin is decreased, then what will happen? It causes the anemia. It causes the right, it causes the anemia. Anemia, that is decreased amount of the RBCs and the hemoglobin. Clear? What will happen? What are the signs and symptoms of the anemia? That is that poor concentration, right? Poor concentration, dyspnea, that is difficulty in breathing, always feeling letharginess, right? Not feeling properly. So all these things are comes under the symptoms of the anemia. Pale yellow skin color, right? If the amount of RBCs decreased abnormally, right? If the amount of RBCs decreased abnormally, then it is called the erythrocytopenia. And if the amount of RBCs increased abnormally, then it is called the erythrocytosis, right? Erythrocytosis. Clear? So this is a short summary of the RBCs, right, which is important for us to crack the questions. Now come to the WBCs, which is also known as the leukocytes, right? Leuko, when we split that word leuko that means white, whitish, and cytes, that is cells. So they are the white color cells. They are the white color cells. That's why the full form of WBC is the white blood corpuscles, right? White blood corpuscles or white blood cells. WBCs makes our immune system strong. Actually it makes a defense mechanism of our body, right? These WBCs makes a defense mechanism of our body. They are the nucleated cells, right? They are the nucleated cells devoid of hemoglobin. That is, hemoglobin is absent, that's why it is colorless, right? That's why it is colorless. WBCs are of two types. Granulocytes agranulocytes, right? Granulocytes and agranulocytes. What will happen in granulocytes? Granulocytes, it shows staining actually. They are of three types. Granulocytes are of three types. First one is the neutrophil, right? First one is the neutrophil. Second is eosinophil, and third is basophil. First is neutrophil, second is eosinophil, third one is the basophil, right? Neutrophil, wait, and in agranulocyte, monocytes and lymphocytes. Clear? Neutrophil 60 to 65%, eosinophil 2 to 3%, basophil 0.5 to 1%, monocytes 6 to 8%, lymphocyte 20 to 25%. Clear? Now what will happen? Neutrophil's nucleus is, it is 3 to 5 lobed actually, right? It is 3 to 5 lobed, like that. Eosinophil, bilobed, or we can say spectacle shape, right? Bilobed or spectacle shape. Basophils, trilobed or S-shaped nucleus. Trilobed or S-shaped nucleus. Clear? Monocytes, large, it's kidney shaped, right? It is kidney shaped nucleus, and lymphocytes large oval shaped nucleus, right? Large oval shaped nucleus. Maximum in amount, neutrophil, basophil, minimum in amount, right? Monocytes, largest WBCs, right? Monocytes are the largest WBCs. Clear? 20 to 25% lymphocytes. So this is the classification of the WBCs, and actually they acts as the neutrophil and monocytes acts as a phagocytic cell, right? Both acts as a phagocytic cells or helps in the phagocytosis, right? They helps in the phagocytosis. Clear? Eosinophil acts in the in case of the allergy. Basophil when some foreign particle enters inside the body, then it acts as a mast cell also, right? And secretes the histamine, heparin and serotonin. B lymphocyte, that lymphocyte is divided into two, that is B and T lymphocyte, right? B and T lymphocyte. Clear? And now that T lymphocyte further divided into three, that is T helper, T suppressor and T killer. T helper, T killer, T suppressor. Clear? So this is the division of the WBCs. Clear? Now come to the normal count of WBCs, that is 6,000 to 8,000 per cubic mm of blood, right? Normal count of WBCs, that is 6,000 to 8,000 per cubic mm of blood. In basophils, basophil secretes three kinds of substances: histamine, heparin and serotonin, right? Histamine, heparin and serotonin. Histamine is also known as the vasodilator. Serotonin is the vasoconstrictor. Heparin is the natural anticoagulant. Right? Heparin is the natural anticoagulant. Now what will happen?